Thanks for joining us on another exciting journey into the world of science, right here on the Sand Science Experience. If you enjoy this kind of content, hit like and subscribe. It's the best way to support the channel and keep these episodes coming. Today we're plunging into something absolutely fundamental to how our bodies work, the tiny powerhouses within our cells called mitochondria. And we're going to be focusing on a fascinating new study in mice that shines a light on a specific protein, SMIM20, and its critical role.
That's right. Mitochondria are truly the engines of our cells. You know, they don't just generate energy, they're also heavily involved in metabolism and, well, signaling within the cell.
Communication, yeah.
Exactly. And when these engines misfire, it can ripple through our health. And this is particularly true for organs that need a lot of energy, like the heart.
Makes sense. And to really get under the hood of this, we've got a scientific article detailing how researchers investigated the function of this protein called SMIM20 in mice. So our mission today is to pull out the key insights, understand why SMIM20s seem so important, and what happens to the heart when it's not functioning correctly, at least, in these mouse models.
And there's a key insight right off the bat. What they found in the heart was, well, quite unexpected.
Intriguing. Okay, let's start with the basics then. Mitochondria and energy.
Okay, so mitochondria produce most of our cellular energy, ATP, through a process called oxidative phosphorylation, or OXPHOS for short.
Right.
It involves a series of protein complexes in the inner mitochondrial membrane. And the final one in this chain, that's complex 4, also known as cytochrome C oxidase.
The finish line for the electrons, basically.
Pretty much. It's absolutely crucial for the whole energy production line.
And complex 4 isn't just a single entity, is it? It's more like a team effort.
Precisely. Complex 4 is actually a sophisticated assembly. It's built from proteins encoded by both the mitochondrial DNA and our nuclear DNA.
Ah, so two different genomes contributing.
Exactly. And to get all these pieces put together correctly, the cell uses what we call assembly factors. Think of them as specialized construction workers for these tiny machines.
Got it.
Now, this study zooms in on SMIM20, which also goes by the name MICa7. It acts as a late-stage assembly factor specifically for complex 4.
Okay, late stage. So it comes in near the end of the building process.
That's the idea. Now, what makes SMIM20 particularly interesting to study?
Yeah, are there known problems with it in people?
Well, here's the fascinating part. We haven't actually identified major mutations in the SMIM20 gene in the general human population yet.
Okay.
But researchers have noticed that reduced levels of this protein seem to be linked to an increased risk of heart failure.
Ah, so even just having less of it might be problematic.
It strongly hints that even subtle issues with SMIM20 could have pretty significant consequences for heart performance. So, to really nail down its role, these researchers used a powerful tool.
Let me guess.
CRISPR.
You got it. CRISPR-Cas9 gene editing. They generated mice that completely lacked the SMIM20 protein. These are the SMIM20 mice.
And what happened? Were the mice okay?
Well, here's the first surprise. These knockout mice were viable. They were born, looked pretty normal overall, and even had a typical lifespan.
Really? No obvious major problems?
Not huge ones, no.
Weird.
There was a little temporary dip in weight for the female knockouts around 10 weeks, but that evened out later. So outwardly, you might not notice much.
But under the surface, especially in the heart.
That's where the significant effects showed up. And here's where it gets really interesting and counterintuitive.
Okay.
Instead of the heart getting bigger, which often happens in mitochondrial disease models, hypertrophy, the muscle trying to compensate the SMIM20 knockout mice actually had reduced heart weight.
Smaller hearts. That is unexpected. Right. And they also had decreased stroke volume and cardiac output. Basically, their hearts weren't pumping blood as effectively. That's a key insight that really sets this study apart.
Wow. So, smaller, weaker hearts. Were there other biological signs of distress?
Yes, definitely. The researchers looked at molecular markers, Nppa and Nttb. Think of them as biochemical alarm bells the heart sends out when it's stressed.
Okay.
And the levels of these markers were elevated, confirming that the heart muscle in these SMIM20 lacking mice was indeed struggling.
Okay, so the heart is smaller, weaker, and sending out stress signals. They must have then looked at the mitochondria themselves, right?
Yeah.
Did complex 4 take the hit they expected?
They did, and yes, molecular analysis of the heart mitochondria confirmed it. In the SMIM20 knockout mice, there was a significant drop in both the amount and the activity of complex 4.
Okay, so that confirms SMIM20's role pretty clearly.
Absolutely. It nails down its job in building functional complex 4 in the heart. But here's another twist.
There's more.
Oh, yeah. They discovered increased levels and activity of complex 1. Remember, that's the entry point for electrons into the whole OXPHOS chain.
Complex 1 went up, while complex 4 went down.
Exactly. Complexes 2, 3, and 5. They were largely unchanged. So a very specific pattern emerged. Here's another key insight. Losing one component seemed to boost another, which is weird.
That is weird. How did this whole imbalance affect the heart's actual energy generation? Especially since the heart loves fatty acids.
Good question. Since the heart primarily runs on burning fats, fatty acid oxidation, they check that. And as you might expect with the complex 4 problem downstream, fatty acid oxidation was significantly down in the knockout heart mitochondria.
Oh, that part makes sense.
But when they gave the mitochondria different fuels like pyruvate or succinate, which feed into the energy pathway at different spots, respiration was actually unaffected.
Huh. So the bottleneck was specifically with using fats.
It appears so.
Did the cells try to compensate? Find other ways to make energy?
They certainly tried. The researchers saw a compensatory increase in the rate of glycolysis that's breaking down glucose in the heart mitochondria.
Switching fuel sources.
Kind of, yeah. And they also saw activation of AMPK. Think of AMPK as like a cellular fuel gauge. It gets switched on when energy levels are low, and it tries to make the cell more efficient.
So the cell knew it was struggling with fats.
It seems that way. It suggests the heart was trying to shift its metabolism, rely more on glucose to make up the difference. And here's maybe the biggest surprise.
Oh.
Despite all these metabolic shifts and the hit to complex 4, the overall cellular ACP levels were actually maintained.
They kept the energy levels stable. How?
Likely due to this remarkable compensation, the increased glycolysis, maybe other subtle shifts, the cell found a way.
That's incredible resilience. But mitochondrial problems often lead to other nasty stuff, like reactive oxygen species, ROS, those damaging molecules.
That's a critical point. Disruptions in OXPHOS can lead to more ROS. And indeed, in the SMIM20 knockout mice, they found a significant increase in mitochondrial ROS levels.
Okay, so more oxidative stress. Did that tie into the membrane potential? You need that potential difference to make ATP, right?
Right. But interestingly, despite the OXPHOS defects and the increased ROS, the mitochondrial membrane potential itself remained normal.
Normal potential, but more ROS. How does that fit?
Well, the researchers speculate that the increased ROS might actually be linked to that elevated activity of complex 1 they saw.
Ah, the complex that went up might be leaking more ROS.
That's a leading hypothesis. And it's another fascinating contrast because in many other cases of complex 4 deficiency, you often see complex 1 function go down, not up.
So this SMIM20 situation is really carving out its own unique path. Another key insight then. The knock-on effects aren't always predictable.
Precisely. The consequences can vary quite a bit.
Okay, so we have increased ROS. How did that affect the heart cells? ROS can damage things, right?
It sure can. And the study revealed that this elevated ROS seemed to be connected to problems with how the heart muscle cells handle calcium. Calcium is vital for muscle contraction, for the heartbeat itself.
Right. The trigger for contraction. What went wrong?
They found the knockout mice had lower calcium levels, both when the heart muscle was resting, diastole, and when it was contracting, systole. The overall calcium transient, that rapid surge of calcium needed for a strong beat, was reduced.
So a weaker trigger signal.
Essentially, yes. And they even found less calcium stored away in the sarcoplasmic reticulum, the cell's internal calcium tank.
That sounds like it would definitely impair the heart's ability to beat properly.
It certainly could.
Okay.
And they also saw something else, an increased frequency of calcium sparks. These are spontaneous little localized releases of calcium from that storage tank.
Sparks. Are those bad?
They're often linked to cardiac pathologies, yeah. A sign of instability in calcium handling. But here's the crucial experiment connecting ROS and calcium.
Okay.
When they treated these isolated heart muscle cells with MitoTEMPO, that's a compound designed to scavenge mitochondrial ROS, guess what happened?
The sparks went away.
The frequency of the calcium sparks returned to normal.
Wow. Direct link.
It strongly suggests the elevated ROS was directly causing these calcium handling problems. And what's more, treating the isolated mitochondria with MitoTEMPO also improved their ability to burn fatty acids.
So the ROS was messing with calcium and fatty acid burning.
It looks that way. It really highlights how interconnected everything is. The electron transport chain, ROS production, calcium signaling, fuel metabolism.
Okay, so let me try and piece this chain together. Loss of SMIM20 means impaired complex 4 assembly. But unexpectedly, complex 1 activity goes up.
Correct.
This imbalance somehow leads to increased ROS production, maybe from that overactive complex one.
Plausible hypothesis.
The increased ROS then disrupts how the heart muscle cells manage calcium, leading to weaker contractions in those unstable sparks, and also seems to hinder their ability to use fatty acids for energy. It's like a molecular domino effect.
That's an excellent summary. It really captures the flow of events suggested by the study.
And what about the complex 1 increase? Any ideas why that happened?
The researchers propose it might be a compensatory mechanism, an attempt by the cell to maintain that crucial mitochondrial membrane potential, even with complex 4 partially offline.
Trying to push more electrons in at the start to keep the pump primed, maybe?
Something like that. And going back to the heart size, the fact that the membrane potential was normal, combined with that AMPK activation we mentioned earlier.
The fuel gauge.
Right. That might explain why these mice showed reduced heart rate instead of the typical hypertrophy. AMPK activation is known to actually put the brakes on pathways that lead to heart enlargement.
So, multiple unique features converging here. Reduced heart size, maintained ATP, increased complex 1, specific fuel issues, ROS-driven calcium problems, all stemming from this one assembly factor, SMIM20.
Exactly. The main takeaway for me is that SMIM20 is clearly essential for proper complex 4 function in the heart, and its absence in these mice triggers this very specific and somewhat surprising cascade of events.
Yeah, the impaired energy metabolism is expected, right, but the increased ROS linked to complex 1 and the resulting calcium issues. That's the really novel part.
Absolutely. That unexpected interplay between complex 4 deficiency and increased complex 1 activity is a really significant finding. It just underscores how incredibly finely tuned and interconnected these mitochondrial processes are.
And it serves as a good reminder, doesn't it? We can't just assume all mitochondrial dysfunction looks the same.
Definitely not. The precise molecular problem really dictates the specific consequences.
So what are the potential implications for us, for humans?
Well, these discoveries in mice could be really important for understanding some human heart conditions, particularly those cardiomyopathies where the cause isn't clear, but a mitochondrial basis is suspected.
Could SMIM20 issues, maybe subtle ones, be involved in some of those undiagnosed cases?
It's certainly a possibility this research opens up. It gives scientists new avenues to explore, new things to look for when investigating heart problems that might stem from mitochondrial issues. It deepens our understanding of just how diverse these problems can be.
Really fascinating stuff. It shows how looking at one tiny protein can reveal so much about the complex workings of the heart.
It truly does.
Thanks for listening. And as always, don't forget to like the video and subscribe if you haven't already. And here's a final provocative thought for you to ponder. Could seemingly unrelated symptoms like subtle changes in heart size and maybe exercise intolerance, things that aren't dramatic heart failure yet, actually be linked back to these intricate mitochondrial processes we've discussed? What other unexpected connections might be lurking within our cells, just waiting to be discovered?